Guys, this is Ms. Steyer with a screencast for CP Biology. Today's screencast is on Topic 3, Transformation in GMOs. This is screencast 3 of 4 in our Genetic Engineering unit. Before you watch the screencast, you should open up your textbook, take a moment or two to read section 15.3 in your textbook. That'll give you the background information that you need for all of the examples that we used today during our screencast. Also, I've linked 2 videos for you during today's screencast that I'll go over. You should pick one that you feel more interested in, and I would like you to, after you're done the screencast, go find the PowerPoint and click on the one video that you're interested in. I'm going to be asking you questions tomorrow in class about that video. So let's get going. So today's screencast is about cell transformation and GMOs. So you could probably guess that the 2 things are related. Cell transformation is needed to make a GMO, and a GMO is a genetically modified organism. A lot of times when people hear that, they get freaked out and they're like, whoa, 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 I don't want to genetically modify anything. However, the goal of the screencast is just to show you some different sides to that and just to eliminate some of the confusion in the topic of GMOs. So the first thing you need to know is that cell transformation is essentially the process in which an organism's DNA is changed into a new DNA or into a new genome that includes new traits. Otherwise, in another matter of speaking, is we could say that the new genes are added to this organism. When we recombine or put together different pieces of DNA into a new organism, we call it recombinant DNA. So that's DNA from more than one organism. Now, the word transformation is an important one to remember, and that is that you are literally transforming that cell to express the new DNA in that genome that you've just put in there. So down here we have a picture of what's called a bacterial plasmid, and we actually use this big squiggly looking object as a tool, kind of like as a vector in order to convey this new piece of DNA to a new organism. So this orange band here, this is a circular piece of bacterial DNA, and from our cell unit you should remember that prokaryotes like bacteria usually have free-floating DNA, and a lot of times we talked about it being circular. Now what we are able to do is we are able to use restriction enzymes, which was taught about in 2 screencasts earlier, to cut open the bacterial DNA and insert foreign DNA. Now you might say, what do you mean by foreign DNA? That would be whatever piece of DNA that we are looking to insert into, hopefully, our final organism. So we'll go over a couple examples, but for right now, just realize that this is the piece that we're trying to get into our final organism. Now, to give you some examples of what we mean by cell transformation is a lot of times pet shops have kind of used this cell transformation and genetic modified organisms to their advantage, and rightly so. They're pretty smart. What pet shops have been able to do is they've been able to isolate a single protein that's found in jellyfish. And you might notice that a lot of the moon jellyfish like these actually can glow in the dark. So they have a bioluminescent gene that allows them to actually produce their own light. So what scientists have been able to do is they've been able to find that exact gene in the DNA, cut it out, put it into a bacterial plasmid, and then put it into a new organism like these tetra. So you can go to the pet store and you can actually see different versions of tetra that will actually express these glowing genes from the jellyfish, and they would technically be GMOs. So that means that inside one of these tetras, they would have a cell that has undergone cell transformations. So that's why they're expressed there. This whole thing wraps up this whole idea of the central dogma. So you guys did your projects where you learned that DNA is the instructions of our cells, and that DNA is recoded during transcription into RNA, and that RNA goes to proteins, and ultimately the proteins is what makes you who you are and gives you your characteristics. So these tetrafish are actually expressing that protein that makes them glow. Okay, so bacteria plasmids are used to make recombinant DNA. So that's kind of what I showed you a picture of in that first one. In that first picture, the plasmids are loops of DNA in that bacteria, and we use a restriction enzyme to cut out the plasmid from that bacteria, and then we insert the new gene into the plasmid. So here we have a picture of a bacterium. So this would be a singular bacteria. Notice there is no nucleus. There aren't a whole lot of organelles. There's cytoplasm, and we can't see ribosomes, but they would definitely be in there as well. Here is the bacteria's singular chromosome, but a lot of times they'll have a plasmid as well. And a plasmid is just a circular piece of DNA that is used to infect its host. So when a bacteria infects an organism, they will actually put their plasmid into that organism. So you can kind of think of it that the bacteria is the real infiltrator. He is the one that gets in there and puts the plasmid into the organism, and bacteria are actually able to insert their DNA into our own DNA. So what we do is we hijack the bacteria and we say, hey, we know what you do. You must insert your genes into another organism. So we then, being smarter than the bacteria, put our genes into the bacteria, or whatever genes we want to put into an organism. And then we purposely infect that organism with the bacteria that we tinkered with, and then that organism will then take that new bacteria and express the trait. So that same bioluminescent gene that we talked about in the tetra fish has also been applied to baby mice. So believe it or not, there are such things as glow-in-the-dark baby mice, and it's actually been applied to kittens as well. And the first question that everybody asks me whenever I go through this lecture is, have we tried it on humans? Can we have glow-in-the-dark humans? And the answer to that is no, because we deemed a while ago when we first started working with transgenic organisms in the 1980s, we deemed that we did not want to work with human experimentation unless absolutely medically necessary. And as of right now, there is no disease that would be cured by a human glowing in the dark. So we've deemed it as unethical and really not necessary. So, a transgenic organism is the name that we give to anything where one or more genes from a different organism has been inserted into its genome. And here's the important part. It has to also be turned on and expressed, which means that that gene is actually able to be, that gene is actually shown in the genome, okay? So here again is a picture of that foreign DNA. So this would be in the bacterial plasmid. This would be in the organism that we are trying to take from. So this would be like from the original jellyfish. Here would be the bacterial plasmid. So we use restriction enzymes, and we cut right here on the side of the plasmid. And then we take that plasmid and we insert this foreign DNA. So we insert that gene there. And if you remember the other screencast, I went over these sticky end regions here. And then eventually that bacteria will have the plasmid with that new foreign DNA. Now this bacteria we can then use as a tool to go and infect an organism. Okay, so I want to take a little bit of break to talk about the difference between classic and transgenic breeding, because there's a lot of confusion on this, on whether or not GMOs are good or bad, or natural, unnatural, and I think a lot of times it just comes down to the words that are used in the common vocabulary. And I think that science as a general practice is that a lot of times the public fears what they do not understand. So I personally am not for or against GMOs. I think that there are pros and cons, but I do want to make sure that the general public is aware of what they're actually talking about. So let's watch this real quick clip together. It's only about 3 minutes long, and it goes over the differences between classic and transgenic breeding.
Nature made them. The fruits and vegetables we take for granted, scientists say, are not the way nature made them. They have all been genetically modified for our benefit.
People think when they go to the store and buy potatoes or tomatoes or grapes that this is the way they always were in nature. In fact, that's not the case. Here we have a wild variety of potato, in fact, which very much looks like the ancestor of this modern potato. It's the same potato, except for a few genes different that were introduced through breeding and selection. This is the same for tomato, and it really is difficult to believe that using just basic selection and crossbreeding over hundreds of years that you went from this ancestor to modern day tomatoes.
So is GM technology simply a high-tech version of the tampering traditional breeders have been doing for centuries? Everyone agrees there are some significant differences. Classical breeders can only cross related plants, like 2 varieties of potato, and a cross involves mixing tens of thousands of genes at a time. Genetic engineering, by contrast, is much more precise. moving individual genes into plants. And it can also do something traditional breeders have never managed to do: move genes between different life forms, putting not just plant genes into plants, but genes from insects, animals, fish.
You can cross a donkey and a horse in classical breeding. They're very close relatives, and you can get a mule. But you can't cross a donkey and an apple tree in classical breeding.
The modern scientific view of nature holds that all living things, be they plants, animals, insects, are controlled by DNA. All are based on thousands of tiny genes that control the way things grow. Genes that are similar across all species, from humans to the food they eat.
We probably share about 50% of our genes with plants. Take an example. There is a protein cytochrome C, which is a very important component of our respiratory machinery, if you like. And this product, cytochrome C, is identical in you, in a pea, in a cow. It's the absolute same gene. That gene is just a coding sequence. You do not find the whole essence of an organism in that gene.
Scientists argue that since tens of thousands of genes go into making most animals and vegetables, moving a single gene doesn't change the essence of a life form. A tomato with, for example, a single pig vitamin gene is still only a tomato.
Alright, so in that video they went through that a lot of the differences between classic and transgenic breeding is simply just time. That transgenic breeding allows us to skip a lot of the hundreds of years that it takes to manipulate organisms. But in reality, whenever we garden, whenever we raise crops or organisms, we are constantly selecting for traits that we find valuable. So transgenic breeding is not very different from that. It just takes place a lot quicker. So there are a lot of dangers with that. So we need to understand our ecosystems and the effects that that will have on the larger scale of the biology of that area. Also, transgenic breeding allows us to bridge the gap to mate within different species. So we can put a gene from a plant into an organism and vice versa. Okay, so let's get a couple examples of these. So we have transgenic bacteria. These are bacteria that we've actually put a human gene into. So we've spliced a human gene out of a human, we've made copies of it, and we've put it into bacteria. So you might say, what is the purpose of doing that? Why would we ever do that? Well, the reality is we've made bacteria express a gene that we want. So again, what's the purpose? Well, the gene we wanted the bacteria to express was insulin, which is really interesting. We need insulin for people who are diabetic and who cannot synthesize their own insulin. So up to now, we've had to take insulin from pigs, believe it or not. And people with diabetes were dependent upon that pig insulin in order to help them. Well, we have since figured out how to make bacteria express human insulin. So it's exactly the same as insulin that was found in a human pancreas. So it's actually a better remedy to the problem so that we don't have to harvest it from pigs. Same is true for human growth hormone. That's the hormone that actually affects your bones and your muscles, and it tells them how long that you should grow and how tall you get. So we've been able to synthesize human growth hormone in bacteria, harvest it, and then use it to treat different conditions. We've also made transgenic plants. And these are actually very common in agriculture, and a lot of the controversy in GMO products is in agriculture. So transgenic bacteria, you remember we use their plasmid. We put a specific gene into the bacteria, and then we use the bacteria to infect the plant. And the plant, once it has that gene in it, is able to express that gene. So many crops now are genetically modified to express different genes. And one example of that is that we've actually created a type of crop called BT corn. That's what's down here in the picture. This has caused a lot of controversy, and you know, it's good that it has, because BT corn is not actually the best. They didn't quite think that one the whole way through. But BT corn was made so that it actually produces its own insecticide. So scientists were thinking, oh, this is great. Let's splice in this gene. Let's make corn have its own insecticide, because then when the rain comes, it won't wash out all of the insecticides that we spray, all of those chemicals on the fields, and it won't end up in our water systems, which is great. But there was a downside to that. And that is actually one of the options of the videos that you can watch. So if that interests you, you can follow up in that story and click on the PowerPoint that's linked to Schoology, and you can hear about BT corn. Another example is plants that actually produce their own fertilizer and plants that resist freezing. So a while back, tobacco plants are a huge export of the United States. And one of the problems is that we couldn't grow it during certain seasons because it would actually frost too easily. So we found a type of gene that actually allows the tobacco to resist freezing, and that's called luciferase. It's an enzyme that allows tobacco to resist freezing. So it saved a lot of money. We were able to export more tobacco as an export crop, and it helped a lot of farmers out. But a lot of people were a little bit skeptical on whether or not we should really be messing with that. So here's where we get to where you guys are really interested, and that is how can we make transgenic organisms. And here we have two of the most adorable mice. We have a normal mouse over here. He's rather on the chubby side, but not nearly as chubby as his twin over here. And yes, these two are actually twin mice. They are genetically identical except for one gene. And that one gene is the gene that we spliced into this twin's genome. So we put one gene in this mouse and not in this mouse. The gene that we put in there is we actually put in a different version of what we are now dubbing the obesity gene. And what that meant is that this mouse, whatever it ate, it would continue to store as fat, and it would actually make this mouse more obese. So these mice were products of a study that was trying to determine whether or not obesity was genetic. So transgenic organisms are used to study disease and gene function. So there's a lot of application to humans here now. So we've used mice to actually help us study HIV and how our immune system works. So we've made mice actually be infected with HIV, and we've altered their genomes to see whether or not there's something we can do to help with the condition of HIV. We've also bred organisms and used transgenics and GMOs to help create hardier livestock. So that's livestock that's going to be more resistant upon disease. It's going to be larger livestock, healthier livestock, livestock with more muscle mass and things like that. This is a picture to throw in there because this is a bacteria culture plate that involves a bunch of different types of bacteria that we genetically modified to express different bioluminescent colored genes. And they actually painted a really cool picture. All of those smears are actually bacterial colonies. So that line there of the pink is actually one bacterial colony that has divided and divided and divided and divided. And when that bacterial colony is under a fluorescent light, it actually glows pink. They took a different GMO bacterial strain and they put red here, blue, and green, so you can actually see that we've mastered bacterial transformation so much that we can actually paint with it. The real nitty-gritty comes over the concerns for genetic engineering, and I'm sure that you guys have heard some of these before. So we're not quite sure right now of the possible long-term health effects of eating GM foods, and I think that's what a lot of the problem is about. A lot of the controversy here, if you look down here at this picture, it says tomato puree made with genetically modified tomatoes. This is ironic because right now there is not legislation that requires a company to print that the product that they have created includes genetically modified organisms, which means you as a consumer, it becomes very increasingly hard to know whether or not you're eating normal food that has not been genetically altered or GM food. So that's a lot of the controversy. We don't know long-term health effects. So one thing I will say, and a common misconception, is that when you eat a GM food that contains a gene, remember, you do not become what you eat. If you eat bacon, you don't end up bacon the next day. If you eat corn, you don't end up sprouting tassels a week later. If there are genetically modified genes in a GM crop, you obviously are not going to take on any of that gene. So a lot of the people who are worried about the antifreeze protein in the tobacco plants to make them resist freezing, they're really skeptical about using those tobacco plants because they're worried about getting that gene. you guys should know by now that you are not going to become or get the DNA that you eat. We need bacterial or cell transformation for you to get the genes that you would want to be exposed for. So we're not sure the long-term health benefits, but one of them is not that you're going to get the genes that you eat. We're also not sure of GM plants' effect on ecosystems and biodiversity. And this, guys, I'd say it's a huge one. We have been learning that ecosystems are extremely fragile and they're very balanced. So right now, the major concerns with GM products is that we're not sure of the long-term effects on ecosystems and biodiversity, so how many plants and organisms there are in an ecosystem. So this would be one of the cons of GM products, and that is simply that we just haven't had them long enough to do controlled studies and to know what their full effects are going to be. All right, so this is the point where I said that you can kind of stop after you finish the screencast and you can pick one of these two different videos. So I linked to a video about super salmon, and that's a video that goes through how we've been able to genetically modify the growth hormone in salmon to make them almost twice the size. Therefore, farmers that have fisheries that raise salmon can get more product yield and actually increase the productivity of that farm. So if you're interested in that, you may watch Super Salmon. If you're interested in the BT corn that I previously talked about, you can click there. Okay, that ends screencast 3 of 4. Tomorrow in class, we're going to be talking about cloning, so get ready for that. Thanks, guys.